Okay, so good afternoon, ladies and gentlemen. My name is Hartmut Ehrlich. I'm the CEO of a company that Bernard already introduced, Abivax, which is part of the truffle portfolio of companies. Abivax was founded 4.5 years ago based on assets that Truffle Capital had actually been incubating for any time between 5 and 7 years. And in order to attract a really experienced management team and bring the company to IPO, 3 existing companies were merged, bringing their portfolios together and providing the basis for the new company for Abivax to actually get to the stock market. The Euronext in Paris, which we accomplished 18 months after founding the company. And we were able to raise 57.7 million euros, which was, and which still is the largest IPO in biotech at the Euronext in Paris. So Abivax is actually based on 3 core pillars of value. 2 of them related to our front runner or leading molecule, ABX464, which is derived from our antiviral library of more than 1,200 compounds. And based on the antiviral properties of this molecule, this was initially intended to be an antiretroviral, as there are already 25 on the market. But when we looked into the specific properties of this molecule, we saw the potential to actually bring this to the next level of treatment. As you know, HIV over the last 10 to 15 years, based on antiretrovirals, has been turned from a deadly disease to a chronic disease. And bottom line is we are trying to bring the treatment now to the next level, meaning providing a functional cure or a real cure for the patient. We are currently finishing up phase 2A and are planning to go into phase 2B by the end of this year. The same molecule, as it has very potent anti-inflammatory properties, is also at this point in time in a phase 2A study in ulcerative colitis. As you know, this field really screams for additional products for patients. The estimate is that we currently don't have sufficient treatment options for about 50% of the patients with this disease. And actually 1/3 of the patients with ulcerative colitis are ending up with having their colon being taken out with colectomy. And I think this speaks to the medical need of bringing new products into this therapeutic area where the current treatments are not sufficient for many of the patients. And finally, we have an asset, ABX196, which we licensed in from the Scripps Research Institute in La Jolla, in California, which, based on very exciting preclinical data, is now prepared for a study in hepatocellular carcinoma, which of course is a disease that has a specific relevance here in Asia, as hepatitis B infections are running rampant, and unfortunately hepatocellular carcinoma, liver cancer, is one of the consequences in many patients that are dealing with the hepatitis B virus for an extended period of time. So this just shows the experiment that showed us that ABX464 is more than current HIV treatments, and this is a mouse model, a humanized mouse model, where mice are basically total body irradiated to wipe out the bone marrow, transplanted with human umbilical cord blood stem cells. Is there a pointer here? This one? Yes. And then they are being infected with virus. And what you see when you treat with current therapy, the viral load, the virus that you measure in the blood is going down quickly. But when you stop treatment after 50 days, it's coming back, so that 10 to 14 days after you stop the treatment, there are levels of virus in the blood that are like before the treatment. And this is whether you treat a patient for 1 month, 1 year, or even 10 years. When you stop the treatment, the virus predictably comes back in every patient. And this is, of course, a big threat, because not only the patient then can develop full-blown AIDS, but also the patient becomes infective for the environment again. And this is why we are still seeing around 2 million new cases of HIV every year. By contrast, Abivax 464 is actually a compound that is slightly slower in reducing the viral load than triple therapy. But the difference is, once you stop, and this is a logarithmic scale, once you stop treatment in these animals, the virus stays at very low levels, actually 25-fold lower than what you are seeing here. We believe that this is due to the effect that we have on the reservoir. The reservoir is where the virus is hiding during antiretroviral treatment. And we have actually been able, in 2 clinical trials now, with 1 month of treatment, have been able to show that we are able to reduce this reservoir. This is a total of 9 patients in the second study, of up to 50%. We are currently in a study where we are exposing patients for 3 months. Data becoming available in about 4 weeks' time. And then the plan is to go into larger studies, into longer studies, where we can treat patients up to 1 year to really reduce the reservoir to the lowest level possible. I also mentioned to you, and I'm just looking at the watch, I also mentioned to you that Abivax 464 also has very potent anti-inflammatory properties. And when you go into an animal model for inflammatory bowel disease, where you give these mice DSS, dextrane sulfate sodium, which is a potent irritant to the intestine, you see that, and this is an experiment in 8 mice per group, you see that the mice that are just getting DSS, they are losing about 20% of their body weight within 10 days, and they are all dead within 2 weeks. By contrast, Abivax 464, mice have initially a slight reduction of the body weight, but then start to grow again. And importantly, they have a normal intestinal bowel structure, in contrast to these particular mice, where the villi, which is the luminal part of the bowel, which actually is responsible for absorption of liquids and of nutrients, they are completely gone. This is why we have this reduction over here. And in the submucosa, you just see a massive influx of immune cells, which we are preventing with this molecule. We have just completed enrollment of a phase 2A clinical trial, a proof of concept study, for which we are looking forward to present the results in September. Patients are now on treatment until the end of July. Then we need about a month for data cleaning, data management, and stats. And we'll be able to report this in September, with the idea, of course, to start phase 2B then before the year is over. And finally, the last molecule, the Abivax 196, which is of particular relevance to this region. And I can assure you, I was in Shanghai yesterday and on Monday talking to 6 pharma companies. This is a molecule that actually will enhance the treatment effect of checkpoint inhibitor treatment, which is the wave of immune oncology that we are now seeing. But the downside with these molecules is that they are, when it comes to solid tumors, only effective in about 20% of the patients, so 1 in 5. With this molecule, we will be able to boost the response rate because we are actually activating with this agonist the killer cells in the body, the killer cells of the immune system, T lymphocytes, and they are actually preparing the scene then for the checkpoint inhibitors to work. Checkpoint inhibitors are basically tearing down the resistance of the cancer cells to treatment, but you need to have the attackers there, and this is what ABX464 is doing. And it doesn't matter what kind of portfolio you have. What you also need in the company, of course, is the management team. And I realized that I'm out of time. When we put the team together, the requirement was to really get people that have done it before in big pharma. I myself spent all my life in pharmaceutical companies like Lilly, like Sandoz, and then 20 years with Baxter. That's the history for the team, but there's one additional element. People need to be hungry. People need to want to do it again. This is what we have with this team, which is, if you look in the biotech industry, a team that is second to none, at least as I can say for Europe. Thank you very much.